screen sharing. Just a second. Why does it say no PowerPoint available? Just a second. Okay, let me see. I'm trying to share, and this is telling me today, no, my PowerPoint is not seen by it. Why? Okay. Just a second. Why does it say my PowerPoint is not visible? It's open. But if I share my desktop, let us see what happens.
Yes, sir. Now it is visible.
Okay, yes. Can you see my... Okay. Can you see the full screen now?
Yes, sir.
Okay, just a second. I'll take my pointer. Go for my presenter view. Okay. Just to check as usual, left side pointer, can you see? Hello?
Yes, sir.
Top, you can see the pointer.
Yes, sir.
Right side
Yes, sir.
and bottom.
Yes, sir.
Okay, so before we begin, okay, ask your doubt.
So my doubt was regarding Sanger's sequencing method. Sir, in that we use basically we use a primer and then we use a polymerase enzyme so that a new strand is
Correct,
synthesized. So,
correct,
sir, like, and also we are using a DDNTP for the chain
correct,
termination. So,
correct.
like the polymerase, they have this proofreading capacity. So, wouldn't it like they like as soon as the DDNTP has been added, so they will remove that?
See, the proofreading capacity that the polymerase has is to distinguish between whether it added adenine के जगह पे say guanine, okay, or instead of adenine it added something like inosine or xanthosine, okay. So when such errors are made, okay, where instead of adding one base we are adding another base, then that tag polymer or polymerase can realize that it has made a mistake, okay. So for example, normally let us say in the template there is a, then I'm supposed to add a T, right? By mistake I add a C, which I should be adding only if there is a G. Now when I make such type of error, that instead of adding a T I have added a C, then the polymerase is able to correct it, okay. But the polymerase is not able to distinguish between whether at the sugar position there is an extra OH or not, okay. So then that is why you will find that it is unable to correct that error, okay. So if you look at adenine, normal adenine or ATP versus dideoxy ATP, or what you only find is instead of OH there is a H there. That change is undetectable by the polymerase. But polymerase can do proofreading if instead of ATP you put GTP. Okay, are you seeing what I'm trying to tell you? So that proofreading function is when you add a different base, not generally when you add dideoxy instead of D. Okay, now you have to also realize that because it is dideoxy, it doesn't bind exactly the same way to the enzyme as your normal nucleotide. So there is some difference, but probably it doesn't affect the proofreading function. Okay, is that clear?
Yes, sir.
Yeah. Okay. So it'll detect gross differences like A ke badle G, C ke badle T, and so on and so forth. It doesn't catch oxy versus deoxy. Okay.
And sir, what is reptation means?
What does
Reptation means
spelling? Huh?
REPTATION. So it
Where
is
did you
with
read that?
so it was like when we do the gel electrophoresis, and if
Correct.
the DNA fragment which we are separating
Mm-hmm.
in the gel, and if the pore size of the gel is like smaller, but like it's not allowing the passage of the fragment, then
Correct.
basically the fragment they move by a process called reptation.
Okay. I don't know. I'll find out and let you know. Okay.
Yes, sir.
Yeah, I'll find out. Reptation. Okay. It's a term I have not heard before. Okay. But one thing I can tell you, if your pore size is small and your DNA cannot move, then all the DNA will simply rack up on each other at one point, and then because current is still flowing, they'll just push and they'll just, just dhaklofy and go, rather than they'll kind of destroy the gel a little bit and move through. Okay, tear it a little bit, as they say. Okay. Huh? Anything else?
Yes. No, sir.
Okay. So last time, if you remember, we stopped at this point where we had actually looked at initially what was the genetic code, little features of the genetic code. Then we spent some time talking about the tRNA and the activation of tRNA by tRNA synthetases. And then towards the end, we talked about this third, player in protein synthesis, which we call as ribosomes. Okay, and we said that these ribosomes are actually giant enzymes, and they are called giant enzymes because they are composed of a lot of components. For example, if you look at the bacterial ribosome, it is composed of two units which are called as 30S and 50S unit, or what is called as a small ribosomal subunit and large ribosomal subunit, which come together to form a functional 70S unit. Okay, and each of these is very complex. For example, the 30S unit is composed of 21 different proteins, and it is also composed of an RNA which is called as a 16S RNA. Okay. Further, the 50S subunit is composed of close to 36 total proteins. Okay, number of proteins. Variety wise, it is 33. Some of them are present in more than one copy, and it also has an RNA component which is called as 5S RNA and 23S RNA. However, the contribution of RNA towards the overall weight of the ribosome is close to 65%. So ribosomes are said to be RNA dominated entities where proteins actually play a secondary role. Now, the bacterial or prokaryotic ribosomes, we find there are about 15,000 a cell when the cell is actively making protein. They are about 18 nanometers, and their molecular weight is extremely high, 2.7 million. Okay, and the reason we say that the main functional part is the RNA is that ribosomes, when they're doing their main function, that is making peptides, the place where the peptide bond is formed, you do not find any protein near 18 angstroms of that size, which means this function of peptide bond formation is actually catalyzed by RNA, which is present there, and therefore these are referred to as ribozymes. Okay. Now, these RNA pieces, which is 5S, 16S, and 23S, are also very complex structures, and they have a lot of what is called a secondary structure characteristic. There is this hairpin loops and this, base paired areas and loops and stuff like that. So, this is 5S, this is 16S, and this is the 23S. Now, a 16S RNA is also very interesting. It is so unique to a species that many times it is used to actually identify a species. Okay, in microbiology work, no? Sometimes when people are working with microbes and they are trying to isolate new microbes, and they want to show that their microbe has never been in existence before. Nobody has discovered it. They are the first one to spot it. The one thing they need to do is grow that organism, isolate the ribosomes, separate the 16S RNA, and then they have to sequence the 16S RNA and make sure that this sequence is not found in the database. Okay, because if it is, then what you have is a bacteria which is already known. Okay, so and the 16S RNA is so unique; it's like a fingerprint. No species, no two species have the same 16S RNA. Okay, so it's very unique thing, but it is part of this ribosome. Okay. And if you remember, we talked about the fact that this is the 50S ribosome, this is the 30S ribosome, where all these regions which are shown in say gray or green all indicate RNA, and this worm-like things are all indicating the protein which is present in this subunit. And this is where those, if you remember your A site, P site, when we talk about ribosomes, they are located here. And when these two things come together, you get the functional 50S unit. And this is the cleft where the RNA goes in. Okay, and RNA kind of slides in there, and then the codon is read and everything functions appropriately during translation. Okay, and just to compare and contrast, just to keep our things in perspective, the difference between the bacterial ribosome and the eukaryotic ribosome is bacterial ribosome is slightly smaller. It is 70S relative to 80S. Okay. Also, it is smaller in terms of molecular weight. Molecular weight is about 2.7 million. This is about 4.2 million. Each of these ribosomes are made up of smaller subunits. In case of bacteria, 30S and 50S come together to give you 70S. And here is the individual composition that this 30S subunit has got 16S RNA and 21 proteins. This has got 5S and 23S RNA and 36 proteins. If you look at eukaryotic systems, they have bigger RNA, in this case 18S and 33 proteins. And here there is a 5S RNA, 28S RNA, 5.8S RNA and 49 proteins. Okay, so they are much bigger than their bacterial counterparts, and they come together to form the functional ATS subunit. Okay. Now the first evidence that ribosomes are the ones who are responsible for protein synthesis was a very interesting experiment which was done, where they found that if you take ribosomes and if you take short sequences, in this case say trinucleotides, or let us say even polynucleotides which have the identical sequence, UUU, and then to this mixture of ribosomes and these nucleotides, you add all the tRNAs. Okay, what you find is very specifically only one tRNA binds to form a ternary complex. For example, if the ribosome has bound UUU, then among all the tRNAs which are in solution, only the phenyl tRNA will come and bind to this complex, indicating some recognition between this phenyl tRNA, this trinucleotide, and the ribosome. Likewise, if you take ribosome and this RNA piece, or what you call as trinucleotide or polynucleotide pairs, which is all AAA, then when you put all the mixture of tRNAs, you will find only the lysine tRNA goes and binds to this entity. Okay, and that is how people found there is something called as a codon, and this codon helps the interaction between mRNA and ribosome and tRNA, so that everything is done correctly when it comes to protein synthesis. Okay, so this work was also done by the same gentleman who did the genetic code, Nirenberg. Okay, but this one was with Leder. Okay, now once people realize all these things are all together, then in order to understand how exactly things happen, several experiments were done, and that has resulted in our understanding as of today as to how actually protein synthesis occurs. Okay, so protein synthesis occurs basically through what are called as 5 steps. Okay, I've listed here 4 steps. The steps being initiation, elongation, termination and release, and then folding and post-translational modification. The step which I have omitted and which we have kind of talked about is a step which comes before initiation. And the step which comes before initiation is synthesis of acyl tRNAs, where we have tRNAs which are all loaded with the correct amino acids. That step has to be done initially only before we even start the protein synthesis. So even though I don't mention it here because we already talked about it, we actually have spent time talking about this stage only. Little bit about when you said about genetic code and tRNA, we actually were kind of preparing ourselves for protein synthesis. Okay, so we'll begin with the first step, which is called as initiation. Okay, and what I'll do is I'll emphasize more on the prokaryotic system because that is what we understand a little bit more. Eukaryotic systems generally always tend to be little more complex and involve more players than the prokaryotic system. Okay, so with that background, that eukaryote will always be a little more complex than prokaryotes, let us look at the process of initiation. Okay, so in terms of how proteins are made in our body, you'll always find that protein synthesis occurs in our body in the N to C direction, which means that when a protein is made in our body, the first amino acid which is recruited is the amino acid which will be the N terminal, and then new amino acids are added to this amino acid in the N to C direction, which means that the first amino acid is, let us say, for argument, glycine. So that amino acid will be NH2CH2COOH. Now the second amino acid, let us say it is alanine, which is NH2CHCH3 and COOH, that will be added to the first one such that the carboxyl group of glycine forms a peptide bond with the amino group of alanine, which means I am adding the new amino acid to the carboxy group of the first amino acid. And that is why we say that protein synthesis is occurring from the N terminal to the C terminal end. And when the last amino acid is added, what you will find is, in that case, that last amino acid's amino group has formed a peptide bond with the previous amino acid, but its carboxy group is free, and that we call as the carboxyl end of the protein. Okay. What you also find, whether it is kind of prokaryotes or eukaryotes, that the first amino acid that is coded for in the mRNA, okay, which is in bacteria called mRNA. In case of eukaryotes, we call it mature mRNA. Okay, the first amino acid that is added as part of the protein synthesis is always triggered by this initiation codon, which is AUG, and which codes for methionine. Okay. Now, as far as this methionine is concerned, it's very interesting. This amino acid, methionine, has got only one codon, okay, that is AUG, but it has got two RNAs which can recognize this same codon. Okay, and these two RNAs which code or recognize the same codon, which is called as AUG codon, are called as formyl methionine tRNA and regular methionine tRNA, and designated as fmet tRNA or simply met tRNA. Okay. Now, the difference is this tRNA, okay, to which is always added formyl methionine, that tRNA will always bind to the AUG, which is the first codon of the mRNA, what we call as the initiation codon. If this sequence AUG occurs anywhere else in the mRNA, then at that sequence, the tRNA which will come and bind will always be this tRNA which carries only methionine and not formyl methionine. Okay, so the idea is this: the start codon always binds the tRNA which is carrying formyl methionine, and all the internal AUGs, wherever they occur, they will be recognized, and to that will bind tRNA which carries just methionine and not formyl methionine. Okay. Now, what is the difference between the two? Here is what is shown. Methionine is NH2 CHCO OH CH2 CH2 SCH3, and N-formyl methionine has one of the hydrogens remaining in the methionine group, and this is NH2 CH2
So you aren't audible.
Hello, hello.
Yes, sir.
Yeah,
So
I'm on hotspot. There was just current guy either. Okay, can you see my screen? Uh, I have to reshare, no? Let me share it again, okay? Okay, can you see my screen now?
Yes, sir.
Hello? Yeah, okay. So, as we were saying, that in case of initiation, the amino acid methionine, which is usually the amino acid which is added first in every protein that is made, because the initiation codon is a AUG, that methionine in case of prokaryotes is always not regular methionine but formyl methionine, which is coded for by a special tRNA, which only binds to the AUG, which is the initiation codon. Likewise, methionine is also carried by another tRNA, which also recognizes AUG, but it only recognizes AUG when it is present in the middle of the mRNA. Okay, and this is the structure of that formyl methionine. Okay, now see how the process begins. Okay, the process begins in this fashion. The 30S unit, which is what is called as a small ribosomal unit, uh, is initially recruited, and to that two factors which are called as initiation factors 1 and initiation factor 3 come and bind. Okay, and as this binding occurs, the mRNA, which is ready to be translated, is also recruited, and they all form a complex. Okay, now, and the way the complex is formed is that after this mRNA comes and sits and interacts with this 30S subunit, the ribosome actually scans the mRNA such that the AUG, which is the codon which is coding for what we call as the initiating formyl methionine, is placed at a location which is referred to as the P site. Okay, and what is called as peptidyl site. And as that happens, this elongation factor also comes in and sits in what is called as the A site. Okay, so now I have initiation factor at the A site, initiation factor 1 at the A site, AUG which is placed where the P site is supposed to be, which is part of the what you call as the mRNA, and this initiation factor 3, its job is merely to make sure that 30S and your 50S don't come together before all of these things occur. So it is actually like a little initiation factor that doesn't allow 50S subunit to come anywhere near this till all the things are ready for it. Okay. Now, how does this ribosome correctly recognize that this is the AUG which should be placed on the P site and not any AUG which is present in the RNA? And that guiding is done by what is referred to as the Shine-Dalgarno sequence. Okay, all mRNAs in what you call as prokaryotic systems have this unique sequence, which is, if you look very carefully, kind of purine-rich sequence as it is called. And this sequence is always occurring a few bases before the AUG, what you call as the initiation codon. Now, this sequence here is always so unique to a given species that this sequence is recognized by the 16S RNA of the same species. And 16S RNA, if you remember, is present in the 30S subunit. Okay, so if you look at this subunit here, somewhere here is also the 16S RNA. And this 16S RNA interacts with the mRNA in such a fashion that the Shine-Dalgarno sequence is recognized by the 16S RNA, and once that recognition occurs, it searches for the first AUG near that sequence, and that AUG is then placed at what is referred to as a P site. Okay. Now this is called as the Shine-Dalgarno sequence because the two scientists who actually discovered the sequence, and the fact that the sequence always occurs very systematically, a few bases, generally as I say 15 to 13 bases from this AUG, what you call as the initiation codon. Okay, so the way the mRNA is placed correctly is because of the fact that this tRNA here, which is part of this subunit, recognizes the Shine-Dalgarno sequence which is present somewhere here. Okay, so now once that AUG is placed on that P site correctly, then the initiation factor 2, which has a bound GDP, comes in, and it also recruits this FMET tRNA. Okay, what is FMET tRNA? This tRNA which is loaded with formyl methionine, and it brings it all and it makes it bind perfectly to this AUG start codon by this complementary base pair which is present in the anticodon region. Okay, now as this occurs and this placement occurs, this initiation factor ka GTP gets hydrolyzed to GDP plus phosphate, and when that occurs, all of these initiation factors simply leave this setup, and the 50S subunit comes in and clamps down, and you get this functional initiation complex, which is actually signaling the start of what is called as protein synthesis. So see what has happened when initiation is complete. The 30S and 50S has come together. The formyl methionine carrying tRNA is interacting with AUG of the mRNA, and this AUG is a start codon, and it is placed like this because the Shine-Dalgarno sequence here interacts with the 16S RNA which is present in this small subunit. Also notice that this first tRNA, which is called as formyl methionine tRNA, comes and binds directly to the P site, and this is very unusual. This is the only tRNA which comes and goes directly to the P site. Every other tRNA which is involved in protein synthesis first comes to the A site and then goes to the P site. Okay, so this is an exception to the rule. The initiating tRNA, as it is called, which carries formyl methionine, is the only tRNA which comes and directly interacts with what is called as a P site, or sits on what is called as a P site. Okay, as soon as this occurs, now you are set for actually doing the protein synthesis. Okay, and just before we go further, okay, similar to what happens in case of what I call as prokaryotic systems, in eukaryotic systems also these factors come together. Okay, so if you look at your eukaryotic RNA, remember when we made the RNA, we put a 5' cap, we removed all the introns, then we put a poly A tail, then we did some methylation here. All that is very important because this is all recognized by what is called as the 40S ribosomal unit, which is a small ribosomal unit in case of eukaryotes. And what happens there also is something very similar. These things, okay, this poly A tail, this cap, these are all recognized by the 40S subunit, and that allows that mRNA to be correctly positioned so that this AUG is in the P site again. Okay, so here it's slightly different because in eukaryotic RNA there are a lot more signals that this cap and this poly A tail, it makes the job of this RNA a little easier. Okay, there is no need for a shine Dalgarno sequence and stuff like that. What it simply does is it looks for this cap, it looks for the poly A tail that tells it the mRNA is mature. Then relative to the 5' cap, you'll just go and search for the first AUG, and that will be the initiating AUG. Okay, and again in that case, as soon as this interaction occurs, all the subunits come together to give you the functional initiation complex. Okay, so the difference is this: if you look here, in bacteria we talked about only 3 initiation factors. In eukaryotes we have 6 of them. Okay, and how are they distinguished? You just put a small e in front. Small e stands for eukaryotic, and then IF stands for initiation factor. Okay, so there's a whole bunch of them. Okay. Now, once that is done, we go to the next step, and the next step is what we call as elongation. Okay. Now, see what happens in this step. In elongation, the second amino acid tRNA will come. Okay, the second means the one which is the first amino acid after the AUG or the methionine. In case of prokaryotes, it is formyl methionine. In case of eukaryotes, it is simply methionine. So this amino acid tRNA, which is actually the second amino acid tRNA, which codes for the second amino acid of the peptide chain, will come. And then this will be attached, and the amino acid which is carried by this tRNA and the amino acid carried by the first tRNA, which is the formyl methionine tRNA, they'll both come together to form a peptide bond to give you what is called a dipeptide. And this activity is referred to as a peptidyl transferase activity. Okay, and after this is done, the entire thing will what is called as translocate. Okay, rather than me telling you this, it is much easier to see it in terms of a picture. Okay, so here we have this initiation complex. If you remember, ready, 30S subunit, 50S subunit, collectively it is a 70S subunit. This is the mRNA. This is the AUG, which is positioned on the P site. UAG is the anticodon to AUG. That is the anticodon in the tRNA, which is carrying formyl methionine. That has come and sat here. Now see what happens. Based on whatever is the codon sequence here, the corresponding tRNA, which is carrying the appropriate protein. In this case, I will call it amino acid 2. Okay, because this is amino acid 1. That tRNA will come and position itself at what is called as the A site. Okay, so at the A site now there's another tRNA, and this tRNA is the one whose anticodon loop perfectly is complementary to this one, or wobble wise is complementary to this one, and it is carrying the appropriate amino acid. Okay, as soon as these two have come together, then the next reaction occurs, and the next reaction is what we call as the peptide bond formation. Okay, now remember, on this P side is formyl methionine. Okay, and how is that formyl methionine attached? If you remember, it is attached via the 3' OH of the ribose of the last 3. If you remember, I told you it is a CCA always. So it is attached to the A ribose and it is attached through ester bond. So you have OCO then CH, and then NHCOH. This is the formyl part. Okay, this R here is for the methionine side chain. Now, sitting right next to it is another tRNA which also carries a amino acid, and that amino acid is OCO, CH, then R2, whatever that R2 is, and NH2. Now, this amino group is actually free, it is nucleophilic, and what the electrons do is they attack this carbonyl carbon. When they attack the carbonyl carbon, I form a bond between NH and this carbonyl group, which is called as a peptide bond or the amide bond. At the same time, this O tRNA is removed as a leaving group. And essentially what happens now is that this formyl methionine gets attached to the tRNA which was carrying the second amino acid. Okay, so think of this. This is one F met, this is the second, and now this amino acid is going to get attached to this, and that is shown here. Okay, in terms of how it looks, structure wise, this is OCO CH R2 of the second amino acid, then NHCO CH R1 of methionine, then NHCHO. Okay, so at this point, see what has happened. The formyl methionine, which was sitting on the tRNA, which was on the P side, has been transferred to the tRNA which carried the second amino acid and which is sitting on the A side. Okay, so in essence now, this initiating tRNA has given methionine to the second tRNA, and it is attached to that via peptide bond. So now this is carrying a dipeptide. Okay, as soon as this happens, something beautiful happens now. Okay, so see what happens now. This is where we are. This is the deacylated tRNA, and now the ribosome moves. And when the ribosome moves, something very interesting happens. This initiating tRNA, which initially carried methionine or formal methionine, now moves to the E side. And E side, you can call it as an empty side if you want. Okay, and then this tRNA, which now carries a dipeptide, moves to the P side, which makes the A side now vacant for the tRNA, which is carrying the third amino acid. As far as the codon is concerned, that can come in, and that will come in here. Again, a peptide bond will be formed where these two amino acids will be transferred to this. Again, this will shift, and so on and so forth. And this process will keep on occurring time after time, and every time the ribosome will move along the mRNA, and it will keep on going. And as it keeps on going, 3 bases at a time will keep on adding one amino acid. Now the beautiful thing is, the last tRNA which has arrived will always carry the entire peptide chain at its end. Okay, because you can think of this, no? This came second. When it came second, it took the first amino acid onto itself. This has come third. When it came third, it took the 2 previous one onto itself. Like that, if the 10th one comes, it will take the previous 9 amino acid containing peptide and take it onto itself. Okay, and that is how this elongation process will occur. Okay, and this will keep on going till you reach a point where you reach the terminating codon, or what is called as a codon, for which technically there are no tRNAs which can carry any protein. Now, as soon as that site is encountered and that sits in the A region, these RFs, what are called as release factors or termination factors, they come in and they have a specific affinity for this UAG codon which is sitting in the A side. And when they come and sit in there, what they do is they enhance the hydrolysis of this bond here. Okay, remember this bond is OCO, right? And this is carrying the entire peptide which was formed till that. So that OCO bond is hydrolyzed, and it is hydrolyzed such that the COH will be released and this will go back to OH. And as soon as that happens, I've got my peptide whose synthesis is over. Now the ribosome carries only this last tRNA which is now empty, and then it has a release factor, and what this release factor will do is allow all of these to separate, and this small subunit, the large subunit, the empty tRNA, RF will all disappear, and all of them can now start to do their job again. So these two will go to a new mRNA and will start their synthesis over. Okay, is that clear? So the bottom line is this: if you think very carefully, to make proteins is not cheap. Each amino acyl tRNA synthesis requires 2 ATP, because if you remember that reaction, the reaction occurs with the release of pyrophosphate, which gets hydrolyzed. Now, every time amino acyl tRNA comes in and sits in what is referred to as the A site, that process of bringing the amino acid tRNA to the A site requires 1 GTP and the subsequent peptide bond formation. And then when the ribosomes move along the mRNA to go to the next codon, which is called as translocation, that also requires 1 GTP. So in essence, 4 GTPs are used to make 1 peptide bond. Now if you remember the free energy for ATP hydrolysis, it basically means I'm putting in 122 kilojoules per mole to form a peptide bond. Now if I hydrolyze the peptide bond, the energy which is released is only 21, which means in a sense I'm investing close to 100 joules in making a peptide bond, and that is kind of a loss in a sense, right? Because when I hydrolyze, I'm going to get only energy equal to 21. But the reason for me willing to spend this 100 kilojoules per mole is the energy for generating information. Because if you think very carefully, I'm not joining any 2 amino acids at my whim. I'm actually joining amino acids very systematically. And I'm forming a peptide which has got a very specific sequence. And I know that the specific sequence is related to a function of that peptide. So I'm actually generating information. So this cost of 100 kilojoules per mole every time I form a peptide bond is a cost I'm paying for so that I can generate good information. Okay. Now, when this synthesis does occur, what you have to realize is when mRNA is being read by ribosomes, as soon as the ribosome starts moving along the mRNA and reaches a certain point, another ribosome can start reading the mRNA again. Okay. In fact, that is very, very common to find that the same mRNA is being read by many ribosomes. And the way you can figure that out is this ribosome, which started from here, will have a protein coming out which will be the longest relative to the second ribosome, which will be slightly shorter, and so on and so forth. In fact, if you take an electron micrograph of a cell where protein synthesis is occurring very actively, you see pictures of this type. Okay, so this is the mRNA, and these are all ribosomes which are sitting on the mRNA. And if you notice, this ribosome which has moved the farthest as a protein which is coming out, which is the longest. Okay, so and that is what is kind of shown here. So if I'm a ribosome and I'm going along the mRNA, reading codons, I'm making bigger and bigger and bigger and proteins, and that protein will start coming out from the ribosome, and I can see that. Okay, and in a sense, this is a very beautiful arrangement you will see, and this type of arrangement are referred to as polysomes. When multiple ribosomes are reading the same mRNA, that system is referred to as a polysomal system. Okay, so so look at here. Here, okay, the way they have set it up is this is DNA. From the DNA, I'm making RNA, and as the RNA is made, I'm making ribosome. Ribosomes are binding, and I'm making protein. Now, notice one thing, that what I just told you can occur only that DNA is made. From DNA you get RNA, and from RNA you get protein. That occurs only in prokaryotes, because in prokaryotes both transcription and translation, they occur in the cytosol. In eukaryotic systems, you'll never find DNA and RNA associated with the ribosomes at the same time, because if you remember, in eukaryotes, RNA synthesis occurs in the nucleus. Then it is matured there, then it comes to the cytosol. And in the cytosol, you might see only this polysome, but you never see DNA associated, because DNA is never seen in the cytosol. Okay? So, to summarize, look at this. in E. coli, protein synthesis, these are the initiation factors, these are the elongation factors, and these are the release factors. Okay. The difference is that if you were to look at E. coli relative to eukaryotes, in eukaryotes, the number of initiation factors, the number of elongation factors, number of release factors are more. Okay. And in the end, both of them are trying to do the same thing, make the correct information rich peptide which can do several functions in our body, like act as an enzyme, act as a receptor, act as a structural protein, and they form the basis of how your cell is actually made up. Okay, so translation is a very important process. It's a very energy consuming process, and it's a lot of energy investment done in order to produce the correct protein and the correct amino acid are joined together so that you get a functional protein. Okay. As I said, just making the protein is not the end. You'll find that both in prokaryotic systems and in eukaryotic systems, after the protein is made, modifications are done to that protein. Okay. Uh, the level of such modifications after translation occurs to a greater extent in eukaryotes. Okay, but it is not something which occurs only in eukaryotes. In prokaryote also modification occurs after translation, as they say, but to a smaller extent. But what are these changes that are observed after the protein is released from the ribosome? Okay, which we call as post-translational modification. Okay, so again, the reason they are called post-translational modification is because all of these changes that we are going to talk about to the protein before they start, their activities occurs after they are released from the ribosome and before they do their functions. Okay, so here is a list of them. There is amino terminal modification, there is carboxy terminal modification, there is what is called as loss of signal sequences, there is modification of amino acids, glycosylation, isoprenylation, addition of prosthetic groups, proteolytic processing, disulfide bond formation, and quaternary structure generation. Okay, and all of these processes, as far as proteins are concerned, they all take place after the protein is released from the ribosome, and that's why they are collectively referred to as post-translational modifications. Okay. Now, just to start, get us going, we'll begin with this first two, amino terminal modification and carboxy terminal modification. Okay. Now, if you remember, in prokaryotes, the first amino acid that is added is always formyl methionine. Okay, and in eukaryotes, it's always methionine. But if you look at most of the proteins in our body, you will find that their amino terminus is not methionine, and in bacteria it is not formyl methionine, which means that as soon as the protein is made, one of the things that occurs almost invariably is that this terminal methionine is removed, or the formyl methionine is removed, and generally at this point the amino group is acetylated. Okay, so and it's very common to find amino groups of the N terminal amino acid of protein, so which methionine has been removed or formyl methionine is removed, having a acetyl group. Okay, and the reason for putting that acetyl group is generally it makes that protein a little more stable to what are called as aminopeptidases. These are all enzymes which will start chewing proteins from the amino terminal. Okay. In addition to this, what is also seen is many times 15 to 20 or 15 to 30 amino acids from the N terminal might be simply clipped, okay, or cut out. Okay, and these are generally termed as amino acids, which are formed part of what is called a signal sequence. Okay, and their job is to help the protein get to the location in the cell that it is made for. Okay, for example, when protein synthesis occurs, after this protein synthesis is done, some proteins are required in Golgi apparatus, some proteins have to be sent to the endoplasmic reticulum, some has to be sent to the cell surface, some might be part of vacuoles, some might be part of lysosomes, and so on and so forth. So how are these proteins directed to all of these different places? Generally, that information is present in the initial part of the protein in what are referred to as signal sequences. Okay, and looking at the signal sequences, the systems in our cell will take that protein and direct it to the appropriate location inside the cell. Now, once a protein reaches that location, then that signal sequence is usually removed. Okay, and that is what we mean by removal of additional N-terminal amino acids. In the same way, at the carboxy terminal also, amino acids might be removed as part of the maturation of the protein, or the amino terminal may be esterified. Okay, so the next slide tells you a few of these things. Okay, so normally if it is just NH2 at the end, we call that unmodified. So one of the modifications which I have already seen is formylation, but you can also have acetylation, that is COCH3, or you can have propionylation, which is COCH2CH3, that is propionic acid. Okay, sometimes we have what is called as pyroglutamate formation. Okay, so here we have the terminal amino acid. Okay, and then let us say the terminal amino acid is a glutamine, and that can kind of cyclize, and you get what is called as pyroglutamate formation. Sometimes fatty acids are added. Okay, myristic acid or palmitic acid, C14 and C16 acid. Okay, and they are added either to the N-terminal, what you call it myristylation or palmitylation, or sometimes you can also add it to the sulfur or the thiol group, or to form a thioester. Okay, that is called as S-palmitylation, especially if the terminal amino acid is a cysteine. Sometimes the amino group which is present in the terminus is methylated. So you get monomethylation, dimethylation, sometimes trimethylation, and a charge on the nitrogen. So all of these modifications are seen at the N terminal. Likewise, at the C terminal, you can get methylation, that is esterification, or amidation, where the OH group is replaced by a NH2. Okay, so such changes are very common, and these occur after, as I say, you have formed the protein, and the protein is released from the ribosome. Loss of signal sequences. This is very, very common, as I said, and these are signal sequences which are shown in different proteins which are actually removed. Okay, this is a very common phenomenon where certain sequences are read by your trafficking system in your cell, as it is called. Trafficking means protein trafficking system, which reads the signal sequences and they direct it to different places. Okay, so here is an example of translocation into endoplasmic reticulum directed by amino terminal signal sequences of some eukaryotic proteins. Okay, so here is some of them. Okay, so this is a very common phenomena, but after the protein reaches there, these signals are cleaved. You can see the sites of cleavage shown here. So all these pieces will be removed because their job is over now. The protein has got to where it is. Okay. Now the next one is what we call as modification of amino acids. Okay. What I'm going to do is actually stop here for the day, and the reason is power has not yet come here, and at any time now, usually what will happen is I will lose my even hotspot. Okay, so before I do that, I'm going to stop my sharing, answer the questions, and then we'll continue next time. Okay, is that okay?
Yes, sir.
So I'm stopping sharing here. Okay, I will check whether there is anything in the chat first. Usually there is something. Okay. Let us say. First is I'm sorry for being late. Okay, that's okay. Okay. Why is formal methionine only used for initiation process? Why can't methionine be used? Does F-methionine play a role in the peptide, or is it a template for protein formation? Okay. What is observed is that there is some sort of recognition that occurs, one between tRNA, which is carrying this methionine and the 16S RNA. Okay. Okay. Now, whatever the way we should also look at it is, if I'm going to make a protein, and if I'm going to make a whole bunch of proteins, I need something, a common signal which will tell me that this is how I'm going to start. Okay, and that is the reason for which maybe at some point during evolution you have to choose any one amino acid uh, to be always the fellow who will start things with. Okay, and in this case, evolution has chosen methionine. Okay, and if you think very carefully, methionine is only one of the amino acids for which there is only one tRNA. The other, I think, is, if I remember right, tryptophan. So these are the only two amino acids for which there is only a single tRNA. And in case of methionine, it's very interesting. Even though there are two tRNAs, the actual loading of methionine is done by only one tRNA synthetase. Okay, so tRNA synthetase is very interesting. It adds methionine to both the tRNA. One which is supposed to carry formal methionine, and the other one which carries regular methionine. Now, the tRNA, which is supposed to carry formal methionine, is acted upon by another enzyme called as formal group transferase. And this is a very unusual enzyme. It recognizes the tRNA and the anticodon and specifically only puts formal group on that tRNA which will be recognized as a start codon tRNA. Okay. Now, the two tRNAs, as I told you, have structural differences. Okay, they are both carrying methionine, but structurally they are slightly different. And because of that, only one can sit at the P site. The other one cannot sit at the P site. The other one will always go to the A site first. Remember that statement I made, that except for the tRNA, which carries formal methionine, which can go directly to the P site, every other tRNA always first goes to the A site. So if I were to take the tRNA which carries methionine and the tRNA which carries formal methionine, and I introduce them both to the ribosome, I'll find that the one which is carrying only regular methionine always go to the A site. It will never go to the P site. While the one which carries formal methionine goes correctly to the P site. And for initiating peptide synthesis, I want something which will go directly to the P site, because only then the second one can come to the A site. I can form the peptide bond and the thing can move. Okay. So hopefully somewhere in this connection of how things happen is a need for this formal methionine to have a very, very specific role. Okay. Hopefully I answered your question, Aditi. Okay.
Yes,
The
sir.
second thing is out of curiosity, what happens to mRNA when it is translated? Will it undergo degradation or is it stored for later use? By and large, okay, after the mRNA is read for a few times by ribosomes and you make a number of proteins from the mRNA, it is invariably degraded. Okay, it is never stored for later use. And as this RNA is always fairly unstable, it will break down on its own also if you don't do anything. So Ankit, usually it is degraded. Okay. So Shivam, so always a new tRNA will bind to the A side of the ribosome and then it shifts to the P side of the RNA with the whole peptide chain. Yes, except for the formal tRNA which goes directly to the P side. Every other tRNA will go to the A side. It will then take all the peptide which is formed onto itself and then it will shift to the P side. Okay. Then even though, Ankit, even though the formation of polypeptide chain is well coordinated, is it possible for a wrong amino acid to be incorporated in the chain? Technically, yes. Okay, and technically, yes. Okay, that can happen. It sometimes happens, but because many of these proteins are not something which are inherited, they have their definite lifespan. All that happens is sometimes out of 10 hundreds of protein molecules that are made, one may be potentially defective because it had a wrong amino acid. But that is where it will end. Okay, it will not. Yeah, it is possible. You can still have a wrong amino acid. A question not related to today's topic. How the PCR fragments are sequenced, like tag polymerase fragment, because of which traditional method are sequenced. So is there any method of doing this? The question was how the PCR fragments are sequenced, right? No, no. If you Shivam, can you elaborate on this? What you are trying to ask? Because I'm not too clear what you are trying to say.
So actually, I read this book. It was of biochem, and then they had some different method of doing PCR fragment sequencing. Like they didn't include that Sanger or Gilbert method.
Okay, okay, okay. Okay. It's not a part of what we do, but that comes under this new sequencing technologies. Okay, and there is something called as PCR based sequencing. Okay, maybe at some point, if we get time, we'll talk about it. We talked a little bit when we said our next generation sequencing, but there are also sequencing methods which are PCR based. Okay, which are different from Sanger, but still help you get the sequence. Maybe at some point I'll talk about it if time permits. Uh, so we'll leave it for now. Okay.
Yes,
Uh,
sir. Okay.
we have 30S subunit where one RNA and many proteins are combined to make 30S units. So why wouldn't we have one big protein and one RNA to make uh, 13? That's a good question. Uh, it is it is probably that if you put all your eggs in one basket, no. See what will happen if I have one big protein and that protein is defective, then I will not be able to make protein at all. Or if I have one big RNA and something happens to that RNA, then I can't make the protein at all. I can't do protein synthesis. But if I have a situation where 30 proteins all contribute to the function, and then if one is defective, the effect on the overall activity is not that much. Maybe it just affects the speed or just affects a little bit of the recognition. I would be in a better situation. It's probably just that our body doesn't want to put all its eggs in one basket by making one big protein and one big RNA. Because if something goes wrong, then that organism will simply die because of not being able to make protein. Okay, so I think the reason is and the other thing is many of these small proteins, no, they all have a little function to do. Huh? It is not that they are just sitting there for nothing. And the requirement of doing something is at different locations on the ribosome. And therefore, a bigger protein with so many different activities at different locations is probably more difficult to design. It's much easier to take smaller proteins with different functions and put it together. Okay, it's like for example, no. Let me see if I can give you an example which will make. Uh, it's like Lego pieces. Okay, if I have Lego pieces, what happens is uh, I will be able to take different pieces and make different things with the same pieces. Okay, but if you tell me rather than you playing with Lego, why don't you have all big toys, one of different type? Okay, I can do that, but then the toy will have only that functionality, nothing else. Okay. So I think the way we evolve many times is many proteins which are having certain functions are reused as part of another system. Okay, and you see that. I don't know whether you notice. If you remember your PCA cycle, and if you remember your fatty acid degradation cycle, no, the enzymes which are used are exactly the same. If you remember in fatty acid degradation, you start with fatty acid, then you introduce a double bond. Then after introducing the double bond, you add water across it. Then after adding water across it, you oxidize the OH group to give you this ketone. The same thing happens in the TCA cycle when you go from, if you remember, fumarate, succinate to fumarate to malate. So that sequence, because it works so well, is also used at other places. So, and that is probably the reason there are so many proteins which are making up one. Because each this small protein is known to do certain thing very well and has been recruited as part of the ribosome. Okay, that is one viewpoint I have, Aditi. Maybe if you read somewhere else, somebody might have another viewpoint. But I think the reason is this, that I don't want to put everything in one basket. Uh, Thermodynamics
Yes,
says infinitely small steps lead to maximum work. Yeah, maybe that is also a reason that all of them do little things, and then in the end, together they make a protein. Okay?
Yes, sir. Sir, and during elongation, when we have this big peptide synthesis forming, won't it kind of unstabilize, destabilize the whole complex? Like we are
Yeah, yeah.
continuously adding. So how is it prevented? And during the formation itself, you mentioned during post-translational changes that there is a protein which, not protein exactly, an enzyme which attacks the proteins, and so it is protected. So won't that affect the growing peptide during elongation?
Yeah, yeah. It all of these things can potentially happen. And as you say, the bigger the protein that I'm making, you can imagine the more the load on the tRNA, correct? Because imagine that I am the tRNA, which is the 300th amino acid in a peptide chain. By the time I come into the ribosome, there are already 299 amino acids which are added, and I'm going to take that and put it onto myself. Okay, you are very right. It's something which is not easy to visualize, and imagine taking all that on one tRNA. And sometimes you feel कि इतना बड़ा amino acid peptide एक end में, the tRNA will simply fall off the ribosome because of the weight. But it doesn't happen because it's other proteins which are part of that 50S subunit, hold the proteins in place, okay, and they stabilize it, okay. Because the peptide that you start seeing coming out of the ribosome, you only see it after about 20-30 amino acids are added, okay. So the initial 19 to 20 are actually held inside the ribosome and may kept stable so that the tRNA can still bind correctly, okay. Yeah, and as you are saying, that modification of N-terminal, it is possible that that N-terminal might be destroyed before the modification occurs. Yeah, it can happen. But peptide synthesis occurs very, very fast. Okay, I told you, right? 100 amino acids are added in about 5 seconds. Okay, so if I make peptides which are of anywhere from 300 to 500 amino acids, I'll be doing them in about 15-20 seconds. And remember, when multiple ribosomes are reading, every second I'm probably making a new peptide. And therefore, as soon as they are made, if they are protected, then there is no chance of degradation. Okay? Make sense?
Yes, sir. Yes. Thank you.
Okay, but these are all very fascinating. Okay, there is a kind of like very difficult to answer these things, but you can only visualize and think about them. Okay, maybe this is the reason. Okay, so can I stop recording now? Okay, and
Yes.
today evening, I don't know whether today evening we are going to start kinetics. Okay, and this is like a class for everyone who is interested. Okay, because whether the subject is there or not, it's an interesting class. I don't know whether some of you have signed up for it. I told them to spread the word.
Yes,
If you
sir.
have, so
Yes,
we'll
sir.
begin. First class today is at 4:45. So it's an interesting topic. Hopefully you'll enjoy it. Okay. So, I'm
Yes,
stopping
sir.
the recording.